Hey A&Pers, this is Dr. Jones checking in. This is A&P II Lab 1, Video 4. We're continuing with the ear. So we found on this model the cochlea, which is a spiral that monitors your hearing. So if you cut that open, you can see this structure, and it's this that's just spiraled around and around and around like a cinnamon roll. Cinnamon rolls. Okay, so if you see these structures and this model behind me, you can see that this is one of these giant ugly things. It's kind of ugly. So this is one of these giant heavy things. Let's go with that. Just writ large. So this is these structures, so this is the same thing. So this is a thin slice. If you took a tiny slice of this cochlea and blew it up big. And so this will be the same as what we see in the cochlea slide. So we have different ducts here. This is the vestibular duct. The Latin is scala vestibuli. This is the tympanic duct, scala tympani, and the cochlear duct, the scala media. I always start here with the vestibular membrane because the vestibular duct only touches the vestibular membrane. And then tympanic duct and the cochlear duct. So the vestibular duct and the tympanic duct at the tip of the spiral are actually continuous. So this is the same tube, but folded in half, folded over on itself. So they do have slightly different functions and different locations, so of course they have different names, but they contain the same fluid. They both contain perilymph, which on this model we said was this pink stuff. And then in here, the cochlear duct will be inside the membranous labyrinth. Cochlear duct is membranous labyrinth. So this will contain endolymph, just like inside these blue tubes here. Okay, so we've got vestibular membrane. The basilar membrane will be this kind of gray blue line right here, and it is the base. The tectorial membrane is shaped like a tongue, T for tectorial for tongue. And the spiral organ, which is kind of this region right here, is the area where your hearing happens, where that sensation is received. So the spiral organ, and of course, because this whole tube system rolls around and wraps around and around and around like a cinnamon roll, it is of course a spiral. These cells right here that have the little hairs on them, pull it closer. Let me just zoom in. There we go. There we go. Now you can see the hair cells of the spiral organ, the inner and outer. And then the somas of these neurons that receive the information, zooming back out, are over here in the spiral ganglion. You may recall a ganglion is a clump of neuron somas, cell bodies, in the periphery, and this ganglion spirals around like a cinnamon roll. So this is the spiral ganglion. So how you hear, I already put a couple of my models away. Okay, now we're gonna do it. Let me grab it. Just a second. Okay, get ready for this because this is the cool stuff. So how you hear is your auricles, your pinna, your ears, act as funnels and they catch the sound waves. So sound waves are kind of a crazy thing because all of the sounds that you hear, me talking, the kids, the cat, all of those noises, Lady Gaga, it's all compressions of air molecules, and areas where there's more air molecules, and then another area where there's fewer air molecules, and another compression of air molecules. And these compressions of air molecules move as waves. And that's what sound is. It's these compressions of air molecules, right? So your ears, your auricles, your pinna, are our funnels, and they collect those compressions of air molecules that we call sound waves. and transmit them down your external acoustic meatus, where they then vibrate your tympanic membrane. Attached to the tympanic membrane are the auditory ossicles, first the malleus, and then the incus, and that is, of course, attached to the stapes, and the stapes sits on the round window. So the base of the stapes, when it rocks, it kind of rocks on the oval window, and it makes vibrations in the perilymph that move through these tubes, which then vibrate this basilar membrane up and down, and then bumps the hairs of the hair cells into the tectorial membrane. So it bumps the hairs up into the hair cells. And then that bends the hairs of the hair cells. And in the walls of the hair cells are tactile mechanical receptors. So cell membrane, and then a mechanoreceptor. So when you bend those down, you open up those receptors, and then you change the permeability of the membrane, and ions can cross the membrane. So when you bend the hairs of the hair cells, that changes the transmembrane potential of the hair cell, which changes the neurotransmitter sent to the sensory neuron, which becomes the sensation of hearing. Crazy, right? That's awesome. Okay, what else about ears did we need to cover? The rest are in the slides, so we won't do those here. So this has been A&P II Lab 1 Video 4, wrapping up the ear. The next slide, slide 5, I will show you the endocrine big flat purple model, and the rest of this lab will be on slides that you will need to know. So thanks as always for tuning in. This has been A&P II Lab 1 Video 4. Education must go on. Dr. J out.